This tutorial shows how to sort, select, and search for genes of interest in the Visual Genomics Analysis Studio, VGAS. Genes and the number of cells expressing them are listed in the Genes panel of the RNA-seq Plot View Options window. Genes can be moved around the list using the move up and down arrows. There are multiple ways to search through genes. The first is by using the search bar at the top of the panel. Users can type a gene of interest in using the Hugo gene nomenclature, then, by selecting Ctrl-A to highlight all genes and selecting All, users can return all search results. The relevant genes that have been highlighted are indicated in the bottom corner. Users can move between results using the arrows. Users can also bring all results to the top using the move up and down arrow. Genes can similarly be searched using the gene search at the bottom of the panel. This will suggest genes to search as the user types. VGAS also comes with a set of gene markers that can be used to select and visualize data. These are built into the VGAS Ensemble references file which accompanies the application. New lists of gene markers tailored to the researcher's specific interests can be developed and added to the references Excel file. The default gene markers are included in the Markers menu at the very bottom of the panel. To use these panels to search the genes, select the Gene Markers list for cell types of interest from the drop-down menu, and then right-click on the RNA-seq Plot Viewer Genes panel and select Genes from Markers. This will highlight the gene markers for selected cells of interest. VGAS can also use lists of genes copied from an external source to search through the genes list. The user can simply copy the list and then using the right-click menu, choose Select from Clipboard. Genes can be sorted on the basis of rank, gene name, number of cells expressing, p-value, p-adjust, or fold change using the right-click Sort By function.